Well, hello everybody. This is where we start. Your first real Tegrity lecture. We're going to go really slow for this lecture. It's actually going to be a very short lecture, probably in the neighborhood of 45 minutes to 50 minutes. And even in its content, this is really more to just give you a very broad overview understanding of genetics, where we'll be going, what certain terms mean, and just basically getting you comfortable with the field of genetics as well as getting you comfortable with Tegrity, getting you comfortable with online lectures. Really nothing to stress here. So sit back, enjoy. Certainly use this as an opportunity to write your first reflection for extra credit, as we talked about in class. Also use this as an opportunity to debug the technology, get in touch with me with anything that doesn't seem to be working right for you, etc. So we are going to very quickly overview Chapter 1 from your textbook, which is an introduction to genetics. And in this lecture, we'll start by just saying, really? We have to do this? Why genetics? We'll talk a little bit about the different organisms that are used for genetic research. We call these model organisms, and we'll talk about why when we get there. But what are the living life forms that we use to gather genetic knowledge? We'll go over the history of genetics very, very quickly and very briefly, give you some context of when these things were done and why they were important. Genetics is a pretty large field. Although a subfield of biology, genetics is its own kind of meta field or umbrella field. So we'll discuss some of the more specific, more detail-oriented subfields within genetics, just to give you a sense of what those are. And we will end the lecture with some core fundamental concepts and probably more importantly terms that we will be using all semester. So for many of you, I guarantee, whether you'll admit to it or not, you are asking yourself or your friends or your parents, why in the world do I need to take this course? I already know I'm going to hate this. I've spoken to so many people who hate this. Well, give it a chance. It's actually a hugely fascinating field of study. I've already told you a little bit about myself and the mess that this course got me into as a younger student. But don't judge a book before you even looked at its cover. Give this course a chance. And don't resort to any final fatal statements. No, just hang with me, and I'll guarantee you that we'll make this enjoyable. Believe it or not, genetics is rapidly becoming the backbone of all biomedical and life sciences fields. In fact, you really can't stray into the life sciences without encountering genetics in some way or another. Certainly molecular biology and genetics are so intertwined and entangled, it is often difficult to separate the two fields. But genetics is playing a larger and larger role in neuroscience. We're beginning to see the genetic and inheritable underpinnings of the workings of the human brain. Pharmaceuticals is rapidly becoming a genetics-governed field in both the development of new drugs and the efficacy of those drugs. We'll touch on this a little bit later even in this lecture, but different people respond to drugs differently. We know that. That's common sense. If any of us has had the unfortunate event of ourselves or someone we know and love being afflicted with a medical condition that required drugs, we know that different side effects affect different people in different ways. It now seems that genetics is largely responsible to patient outcomes when treated with pharmaceuticals. Same goes for medicine. How a patient will respond to given medical therapies and treatments is largely governed by their genetic predisposition for those therapies. The development of living things, the cellular physiology of living things is governed quite substantially by the genetics, by the DNA in those organisms. For those of you who are forensics majors, the field of forensic DNA technology is a genetics field. There really is no difference in the core fundamental concepts between forensic DNA technology and genetics. The two are one and the same. Forensic DNA technicians are geneticists. So who we are, and although we can debate this fact philosophically, what we do because of that 6 feet of DNA that is in each of our cells, that 6 feet of DNA that we inherited genetically from our parents. Our DNA is truly the blueprints of who we are. Our DNA makes up our genome. Our genome is responsible for governing the proteins our cells make, and those proteins are the machines that make us who we are. You cannot spend a single moment in the life sciences without encountering genetics either directly or indirectly. So, in granting you all a degree in biology or a life sciences field, including forensics, we, and by we I mean the college, Bay Path, are certifying that you, and by you I mean you, the student, understand the core foundations of biology, that you are new experts in the life sciences, and that foundation in biology would not be complete without genetics, because genetics is woven so intimately in all of the aspects of the life sciences. So what's the net result? What's the take-home message? The take-home message is, as our knowledge of biology continues to grow and our understanding of genetics grows with it, things will come forth from that. Changes will occur not only in our field, but in our entire society. Societal changes, policies. These are government initiatives. These are societal initiatives. Will be directly relevant to genetics. We've already seen that in some way with different medicines that are up and coming, with the stem cell debate, etc. Many of you, whether you choose to believe it now or not, will be the leaders in your fields of tomorrow. And we, the college, the society in general, want you to be well versed in the issues and the knowledge that will be important tomorrow. And genetics certainly falls into that. So societal changes and policies will be directly relevant to genetics. It's important that you'll understand how those changes in society will impact the life sciences, but much more importantly, vice versa. The life sciences and genetics will impact society, and if you're playing a role in that impact, you need to understand the implications. Some of you will be the leaders, policymakers, and deciders of tomorrow. Hopefully, with a core foundation in genetics, you will be able to more effectively make the right decisions in those roles. Plus, in my opinion, it's just a fun subject matter to learn about. So deal with it. You have to take it, and it's going to be great. So what is the role of genetics in biology? Well, the human genome consists of about 20,000 genes. Now, that's 20,000 discrete instructions for building proteins. That's a pretty small amount. I mean, 20,000 sounds like a lot. Say, wow, 20,000. But think of that in real everyday numbers. $20,000 is really not a lot of money. If your starting yearly salary after graduating from college is $20,000 a year, you're actually going to be in some trouble. So 20,000 genes spread across 6 feet of DNA is all that's needed to make you you. That's it. That's astounding. Again, each of these genes is the instructions for building a single protein. That's more of a molecular biology concept, but it's certainly critical for genetics as well. This is the idea of the quote-unquote central dogma of molecular biology. A single gene in the form of DNA is transcribed or copied into a single RNA molecule. That RNA molecule is then translated or used to produce a protein. So one gene in DNA allows a cell to make one protein. And these proteins, when they're built, carry out all of the cellular processes that keep that cell alive, and then, as an extension of that, keep us as individuals alive. So it's the proteins that do the doing of life. It is the proteins that are the machines of life. And it's the DNA that is the instruction manuals for building proteins. So if proteins are responsible for life, and DNA is responsible for building those proteins, DNA is the instruction manual of life. But never forget, it all comes down to proteins. It's the proteins that are the machines that carry out the functions of life. It is the proteins that are essential for life to be possible. It is the proteins that keep us alive. DNA is nothing more than the instruction manual for building those proteins. So when a cell or a gene picks up a single mutation in a single gene, it only affects a single protein. If that protein plays a very small, negligible role in life, that effect of the mutation is going to be negligible or even invisible. However, if that protein plays a very, very critical, all-important process in the cell, that mutation disabling that protein is going to have a life-changing effect. We are who we are. Good, bad, prone to good health, prone to sickness, intelligent, not so intelligent, well-sighted, or nearly blind, because of our genes. And that is the collection of all our genes. The normal, non-mutated, what we would call in genetics, wild-type genes, as well as the mutant genes, and all of us are carrying mutant genes in our genomes, giving us our own distinct, unique properties. Genetics is not a new field. In fact, people have been practicing genetics and genetics research since long, long before they knew what DNA was, long before they knew what genes or genetics were. In fact, long before we even called science science. People, humans, have been practicing genetic research for thousands of years. What is this? Oh, I heard someone. That's an ear of corn, a normal ear of corn. That corn is descendant from these species of corn. This is natural corn. It's small. There are a very limited number of kernels on that corn. This is wild, non-domesticated corn. It was good. It was nutritious. It was easy to grow and cultivate. And so man, thousands and thousands of years, took the corn plants that grew faster, easier, and gave larger ears, and mated those with other corn plants that did the same. And after many, many generations of this selective breeding, this selective mating, man was able to create a variety of corn that grew like this, with a larger ear, more kernels, faster growing, easier to cultivate. And they continued that practice and took only the best, biggest corn-producing plants that gave this, and continued to only mate those selectively until eventually we created the modern-day corn. This modern-day corn, while technically natural in that it's not genetically modified, there's been no biotechnology applied to it, etc., is still a man-made creation of speeding up evolution in its pace and its direction, creating a plant that gave us what we wanted as a food source, not necessarily what nature wanted as an ideal plant. These are human beings thousands of years ago. People with no formal training in genetics, practicing genetics research. These people didn't know what genes were or DNA was. Had you said the term allele to them, they'd hit you in the head with a wooden post. These people were simply following the empirical evidence in front of them, realizing that plants that gave large ears of corn mated with other plants that gave large ears of corn, gave rise to baby plants which gave even larger ears of corn. Simple, common sense approach. We still do it in a non-scientific, non-academic setting. This chart represents the way that chickens have behaved. These are market chickens, chickens that are going to be sold for their meat at the store. How chickens behave from 1920 to the year 2000. Let's start with the green line. The green line here represents the body weight of the average chicken sold at market. In 1920, the average chicken weighed 2 pounds when sold at the market. In the year 2000 and a little bit past, the average chicken sold at the market weighed 5 pounds. That's more than a doubling in the body weight of the average chicken in about an 80-year time span. So 2 pounds in 1920, 5 pounds in the year 2000. That's pretty good. Bigger chickens, cost more money, make more money for the poultry farmer. How long did it take the baby chicken to develop into its full-grown marketable weight? Well, in the year 1920, it took about 17 and a half weeks. Well, I'd say that's about 17 weeks. 17 weeks for a chicken to reach its full-grown size. Now, in 1920, full-grown size was 2 pounds. So in 1920, it took about 17 weeks for a chicken to reach 2 pounds. In the year 2005, more or less, it took about 7 weeks for a chicken to weigh 17 pounds. Think about that. Chickens more than doubled in size and took about 1/3 the amount of time to get to that size. Faster growing chickens growing to a much larger size. How? Recombinant DNA technology? No. Professors and scientists infiltrating the poultry field? No. Poultry farmers realizing that if you took the faster growing fat chickens and only mated them with the other faster growing fat chickens, their little baby chicks were fast growing and fat. And you do that for 80 years and you can very significantly affect the evolution, truly the evolution of these traits in the population of chickens. We human beings are a little bit too smart for our own good, and we breed the foods we want and the foods we eat to fit our demands as a consumer. We breed resistance to pests into our foods. We breed our foods for size, for taste, even for color. Nobody likes green tomatoes. Everyone likes red tomatoes. So we breed the reddest tomatoes we can breed. And again, I remind you, we're not talking about the genetically modified GM, Monsanto kind of revolution to food. We're just talking about selective breeding, taking a naturally occurring process and pushing it, nudging it in the direction we want. Some more examples. Somewhere between 10 and 12,000 years ago in the Middle East, human beings began domesticating crops and animals. Varieties of fruits were developed that never really even existed before through selective breeding, and they were developed based on their taste, their size, and their color. Another example in the Talmud, this is the Jewish book of religious laws. It states that a woman who bears 2 hemophiliac sons, and these are sons who died after circumcision because they bled to death. Their blood was not able to clot for the incisions of the circumcision. Those women would be exempt and waived and wouldn't be allowed to circumcise their 3rd son. Why? Because they knew that if that 3rd son was circumcised, there was a large probability that son would also be hemophiliac and also bleed out. That shows an understanding of inheritance, that hemophilia was an inherited condition, that if the 2 older brothers had it, it's quite likely the younger brother would have it too, and so we avoid that younger brother's death by waiving his circumcision. Even more astounding, the Talmud states that if that happens, 2 hemophiliac sons die, none of the sons of the mother's sister should be circumcised. Are you kidding me? This demonstrates knowledge of what you will learn to be sex-linked traits, traits that are specifically passed from mothers to sons. This is a high-level concept in genetics, and it was understood in the ancient Jewish book of religious laws. How? Through scientific technology? No. Through time travel? No. Through empirical observations. By seeing that it was much more likely for circumcision to travel in one family than across different families. This Jewish law demonstrates an extremely thorough understanding not only of basic genetic principles, but of sex-linked genes and mutations that occur on the X chromosome. Again, you'll learn about this in about 2 weeks. So although early humans understood heredity empirically through their observations, that common sense approach to heredity, they still didn't know the mechanism of inheritance, right? They could see that traits were passed down in families, but they had no idea how. And what genetics, more recent genetics, can show us, explains to us scientifically, is the how genetics works, the mechanism of inheritance. And that's what you'll learn about in this course. Let's fast forward a few hundred or 1,000 years and look at the role genetics plays in modern medicine. Genetics has a huge influence on medicine. I've already alluded to this in the beginning of the lecture, both in the drug discovery domains and the development of drugs. Here's an example. Now, insulin is made recombinantly. It used to be that insulin was harvested from pigs and cows. So human diabetics would actually be taking cow and pig insulin. It was close enough, it did the job. But disease could be transmitted. It wasn't 100% effective because it's not human insulin, etc. Using principles that have been learned through genetic research, we can now make human insulin in a way that's not only easy to produce, but easy to harvest, easy to process, and do so in a much safer, much more controlled environment. Genetics also allows for more personalized diagnoses and more personalized medicine and treatment. The whole movement of personalized medicine comes from our genetic understanding. This is what I was talking about earlier on in the lecture. You have a group of patients. All the patients have the same affliction. All the patients have the same diagnosis. Back in the olden, non-personalized medicine days, if you had a treatment for that condition, you would give it to all the patients and cross your fingers. As the doctor, you would know that some patients wouldn't respond to that treatment and other patients would. In fact, for many treatments, you would also know that some small percentage of your patient pool would have a toxic reaction to the therapy that you were giving them. But what could you do? You had no insight. You didn't know how patients would respond. So you treat the patients for the disease that they have. Now we can screen patients for their genetic profile prior to any treatment at all and categorize patients as very, very highly likely to be good responders to the treatment, or much more likely to either be non-responders or even more dangerously, toxic responders to the treatment. So now the individual patient receives the treatment that's best for him or her. If the genetic profile shows that the patient will respond to that treatment, they receive that treatment. If the genetic profile indicates that the patient likely will not respond to the treatment or have a toxic reaction, the treatment isn't even offered. That is a genetic understanding of patient reaction to treatment, something we couldn't do just 10 or 15 years ago. Amazingly, some genetic disorders, that is, disorders or diseases that are encoded in the DNA, can be diagnosed long before the onset of symptoms even occurs. This is straight through the power of genetics. The best example of this is Huntington's disease. Huntington's disease is caused by what's called an expansion mutation. I won't really define that now because it doesn't add anything to your understanding, but say in a healthy gene that's responsible for Huntington's disease, so in a person who's not going to have Huntington's, in that gene there is a repeating DNA sequence, CAG, 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 and a healthy individual has somewhere between 10 and 26 of these CAG repeats. Huntington's disease is caused by an expansion of these repeats. So instead of having 10 to 26 CAGs, people with Huntington's have 37 to 80. So it's the same sequence, it's just too many of those CAGs, which causes the disease. So theoretically, a baby can be screened for this gene. This gene can be sequenced in an infant. And at infancy, you can tell whether or not this individual will develop Huntington's later in life. If the infant has 70 repeats in their Huntington's gene, that infant will develop Huntington's as an older adult. You should take just a quick moment to ponder all the implications of that. In a very kind of separated, I don't want to say the word heartless, but in a very clinical way, we can say that's amazing. We can diagnose an infant in infancy for a disease that they won't even be getting to show symptoms for until they're in their 50s. It's amazing. Genetically, it's amazing. Technologically, that's amazing. But then in a very human and compassionate way, you have to stop and say, oh my Lord, do we want this? Do we tell the child that this is what's in store for them as they grow? Is this knowledge we should be privy to? How does this affect the psychology of the patient? And so there's a lot, again, of these societal implications, these human considerations that come with these genetic technologies that we develop. It's even more amazing for Huntington's because the number of repeats gives you a strong indication of the onset of the disease. So remember, I said anything more than 26 repeats gives you the disease. So if you have 45 repeats, you're likely, very likely to have Huntington's. But based on that, 45 repeats, we can give you a general ballpark figure that you'll begin showing symptoms of Huntington's between the age of 40 and 45, 40 and 50. More repeats means an earlier onset. A very, very high number of repeats, like 70, gives preteen onset of Huntington's disease. So not only can we diagnose the disease based on the number of repeats, we can also give you, the patient, an idea of when in life you can expect the symptoms to begin. It's almost frightening in a way. Almost frightening. Finally, I'm sure many of you have heard the term gene therapy. This is the idea of finding genes that are mutated in the human genome and then going in and replacing those mutant genes that will cause disease with healthy genes that will not cause disease. Gene therapy is still perhaps one of the largest miracles humans will ever develop, but there is no viable gene therapy available right now. The few times it has been tried in human patients, the outcomes have been mixed, to say the least, diplomatically, from some evidence of successful outcomes to no change in the patient's condition, and I know that one patient has died as a direct result of a gene therapy protocol, and there might have been 2 deaths in that. So still this idea of tinkering with genes and the living human being for the sake of treatment is still a little bit iffy, and still needs many, many years of development before it could ever be used widespread on the human population. So that's, in a nutshell, how genetics plays a role in our own personal health. What else do we know about genetics? Or more, a better way to say that was, what else does genetics tell us and allow us to understand about our own existence as human beings? To me, the most powerful aspect of genetics is that it truly unites us all. And I don't mean us all of us people, although that itself is its own kind of philosophical discussion, that we are all so much the same and so infinitesimally different that the fact that we fight and bicker and find differences amongst each other is laughable. We are so much the same, cut from the same cloth. But in fact, we are so much more alike to all living things on this planet than we are different. Genetics unites life in a very fundamental way, and that's all life, from the simplest bacteria all the way to human beings. Every living thing on this earth uses the same DNA in the same way for the same outcome. And I'll say that again. Every living thing on this earth, from your neighbor to the bacteria you wash off your hands each day, uses DNA in the same way for the same purpose. They use that DNA as an instruction manual for building proteins. Every living thing on this planet uses proteins in the same way for the same purpose. Those proteins allow living things to survive on this hostile planet. And all living things use their DNA to make proteins to survive in roughly the same way. Now, that's not only uniting philosophically, but it also has a very strong scientific implication. By studying how one living thing uses DNA, and remembering that all living things use DNA the same way, we are, in a very real sense, studying all life when we study one life form. Every living thing on this planet has a DNA genome. That's the sum total content of its DNA. Every living thing on this planet uses that DNA genome as an instruction manual for building proteins. How these instructions are implemented to make proteins, processes like transcription and translation, how that DNA is repaired after mutation, how this DNA is copied during cell division, a process called DNA replication, all of those processes are nearly identical in all forms of life on this planet. Without insulting any religious beliefs you may have, scientifically, life evolved from a single ancestor, making all of us cousins, making all of us similar on this planet. A single protoplasmic, gloopy little amoeboid-like, freaky little cell sparked into life billions of years ago. And from that cell descended all of life. We all have the same great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great-great- so we all share so much in common despite all of our differences. So all life on this planet evolved from a single common ancestor. Think about that for a second. There's some implication there. What does that mean? What does that make possible? Well, I already alluded to it in a way. What that means is that when we study other forms of life, we learn more about ourselves. And that leads us into the discussion of model organisms. The other organisms we use are called model organisms, but it's important to keep something in mind. When you're doing health sciences research, when you're doing life sciences research, when you're researching human conditions and using model organisms, you are never, and you should always avoid falling into the trap of, you are never interested in how that model organism works. You are simply using that organism to study how you, the human being, works. There are a select few organisms that scientists tend to use, and in general, we try to use the lowest life form possible. I'll tell you what I mean in just a second. Bacteria is a model organism. The genetics, physiology of bacteria has been very well characterized, and indeed some of the most remarkable and some of the largest milestone findings and discoveries in genetics and in the life sciences have come straight from bacteria. We'll get into those more in cell and molecular biology, but it's important to introduce it as a model organism here. So an example of that is DNA replication, but that's just one of dozens of examples of huge, huge understandings of life sciences that have come from bacteria. Then eukaryotic cellular processes. Well, yeast is a wonderful model organism for that. Actually, Baker's yeast is the model organism I use for my own research. It is a eukaryote, as I'm sure you know. That means it has an enclosed nucleus. It has membrane-bound organelles. So much more similar to our cells than we are to bacteria. But still very, very similar. In fact, yeast cells at the cellular level do everything a yeast cell does almost identically to what a human cell does, and there's about 70 to 75% similarity between humans and yeast in their DNA and in their proteins. Think about that. You're between 70 and 75% the same as yeast, and about 25 to 30% different from yeast, a single-celled organism. Incredible. If you want to go a little bit higher, processes like cell development, how a single fertilized cell becomes a multicellular organism with some body plan and some organ systems, then you can use C. elegans, a microscopic worm. Senses and behavior come about when we look more at more complicated organisms, such as fruit flies, Drosophila melanogaster. Advanced eukaryotic conditions and processes such as cancer, immune, multicellular, multisystem processes, then we're looking at mouse as a model organism. So this is what I mean by the simplest life form possible. If you are a researcher and you are interested in studying some obscure detail of DNA replication, why would you kill hundreds, if not thousands, of mice to learn about that? Why would you work with something as complicated as flies? Why even use yeast when bacteria replicate their DNA in the same exact way our own cells do? Use the lowest life form possible to answer the scientific questions you can answer. However, if you're a cancer researcher, you would be insane to use bacteria. Bacteria don't get cancer. Bacteria don't even have eukaryotic cellular processes. To study cancer, you need to use a mouse. Is it good to kill mice? No. Do scientists want to sacrifice as many lab animals as they can? Of course not. But if you're answering important questions about cancer, you need to work with a model organism that develops cancer the same way we do. You're going to have to work with a mammal. And so you work with the smallest, lowest life form mammal that has good, well-defined genetics, and that happens to be the common house mouse. There are even model organisms for plant research. Arabidopsis thaliana, it's a weed, it's called the roadside mustardy weed, whatever that means, is the model organism of choice for plant research. The genetics and molecular biology of Arabidopsis have been well defined. All of these organisms I'm discussing here have had their genomes sequenced. We know the DNA sequence of the entire genomes of their organisms. The genetics has been well defined. We know how traits are inherited in these organisms, and so they make very good model organisms. We study them because we understand the fundamentals that make them tick, but they allow us to answer questions that we still have about ourselves. Remember, we use these organisms to learn about us, because genetics and evolution tell us that these organisms are our cousins. So what makes an ideal model organism? Well, ideally it would have a short life cycle. We're dealing with genetics, which means we're dealing with matings, and inheritance, which means we're dealing with generations. If we choose a model organism that has a 50-year generation cycle, and it takes 50 years for parents to mate and then have a full-grown offspring, a single scientist can do a single generational experiment. Bacteria divide every 20 minutes. So in an hour, you have gotten yourself 3 generations of bacteria. 3 generations, meaning grandparents, parents, and offspring. So shorter life cycles and faster reproductive cycles make better model organisms, especially for genetics, because we can see that inheritance pattern in times that are suitable for our own. Large numbers of progeny. We'll touch on statistics here and there in this course, for better or for worse, and the larger number of progeny, the larger your sample size. The larger your sample size, the better the results can be trusted. So matings that produce one offspring are bad for genetic studies because it could have been a fluke. Look, these two parents came together and had an albino child. Is that because every time they mate, they would have an albino child? Is that because this particular child picked up a mutation randomly early on in development? But if two parents have 75 kids and all 75 kids are albino, now we know this is a true inherited trait that came about due to the mating of those two parents. So larger number of progenies gives us a better sense of what's fluke, what's chance, what's random, and what's real inheritable genetics. Because these are organisms that are being used for laboratory studies, the organism has to thrive in a laboratory environment. Whales don't make good model organisms because they're too large, and they can't exist in a typical university or industrial lab. So smaller size, obviously, is important. And cheap and easy laboratory housing kind of falls under that. You want something that's small. You want something that's not going to eat too much. You want something that can stay in a cage and still be happy. You want something that you can keep on a university floor. Large organisms don't really fall under that category. So all of the organisms that I listed on the previous slide, you might have noticed, were quite small in size. There are some other commonly used laboratory organisms that are used for genetic studies. The ones I've showed you on the last slide are what we would call the true model organisms, the classical model organisms of the field, but there are others, such as zebrafish. Zebrafish is used in a lot of genetics labs, and quite a lot of interesting and important findings have come from using zebrafish. One of the things that makes zebrafish attractive to many scientists is the eggs of a developing zebrafish embryo are transparent, and so you can watch the fish develop from the single-celled stage all the way to the juvenile that's about to hatch, and so that's useful for developmental studies. Here's a genetic example of knowledge that came from zebrafish. On the left we see a normal zebrafish with its normal striping pattern, and on the right is a mutant zebrafish referred to as the golden mutant. The golden mutant has fewer melanocytes, so it's got less melanin in its pigmented stripes, and it's also a different color. Rather than being yellow, it's a little bit more golden. The golden mutant, again, has fewer melanosomes, fewer melanin-producing proteins, and the gene responsible for that decreased melanin production, a better way to say that is the gene that was mutated in the golden mutant, was the SLC24A5 gene. And zebrafish has that gene. You mutate that gene in a zebrafish, you get a golden mutant. But humans have that gene too. Remember, we're all cousins. We all have much more similarities in our DNA than we have differences. So we have the same gene. So the question became an obvious one. Well, if we're interested in ourselves, and we know that this mutation in the zebrafish reduces melanosomes, what does this mutation in our own genome do? And, quite strikingly, it's responsible for a great deal of the spectrum of complexions we see in human beings. Those of us with the darkest complexions tend to have two wild-type functioning copies of the SLC24A5 gene, making the largest number of melanosomes and giving the deepest pigmentation. Individuals with more of a moderate complexion usually have one mutant version of this gene and the other is functioning. Individuals in our population with the lightest complexion have the fewest melanosomes, the fewest melanin-producing proteins, the fewest melanin-producing cells, and those individuals tend to have two mutant copies of this same gene. So a finding in zebrafish was directly applicable genetically to the human condition. So a couple of kind of discussion questions I just want you to ponder, maybe a little food for thought in your reflection if you'd like to comment on these there. Do you think human beings are model organisms? Why are we model organisms, or why are we not? And also related to that, do you think a horse would make a good model organism? It isn't yet, but would that be a good organism to pursue as a model organism? Why do you think they would, or why do you think they wouldn't? All right, so moving on now kind of to the last phase of the lecture. We'll talk a little bit of the rise of the science of genetics and then end with some of the terminology and fundamental concepts of genetics. Once it was understood that plants reproduce sexually, like many other sexually reproducing life forms on this planet, plants use pollen as their gametes. The sperm of plants is pollen, but still it's a sexual reproductive cycle. It was understood that plant matings could be controlled. If there's a mommy plant and a daddy plant that give rise to a seed and then a baby plant, scientists could control who the mommy plant was, who the daddy plant was, and control the matings between. By selecting which pollen fertilized which mommy plants, scientists could control the matings of plants. This opened the door to just huge experimentation where plant parents were pre-selected and predetermined, plant matings were controlled, and plant progeny were studied for the traits that they inherited from those parents. This is genetics, the study of inheritance. It was Gregor Mendel, the monk and botanist, who performed these scientific experiments. Mendel is the father of genetics. He is the pioneer of classical or Mendelian genetics, and in fact the whole first quarter of the course, once we finish up with mitosis and meiosis, will be spent going over Mendel's discoveries and his rules. Before Mendel, it was known that life was made up of cells. It was also known that inheritance was passed on by sex cells, that sperms and eggs came together to create a new life. It was near the end of Mendel's life that it became accepted that the heritable material of life, whatever that was, because DNA had not yet been discovered, was located in a cell's nucleus. These are very fundamental concepts to us now. They were new in Mendel's time. Question became, what was in the nucleus carrying this genetic information? There were quite a few theories of what was in the nucleus. Pangenesis, genetic information magically traveled from all different parts of the body and converged at the reproductive organs and then were passed to the next generation. That's not true. The inheritance of acquired characteristics. This is the Lamarckian view, the idea that the giraffe gets a long neck by stretching for those high leaves, so the neck gets longer because that giraffe is stretching. But that stretched neck is passed on to the next generation. That acquired trait is passed on to the next generation. Well, that's not right. One of my favorites, preformationism. The idea that inside each sperm, of course the sperm, right, because the father is responsible for everything, inside each sperm was a teeny tiny, little tiny miniature human being that just needed to make it into the mother's egg, and then it hatched from that sperm and grew into a slightly bigger human, a slightly bigger human, a slightly bigger human. It's laughable now, but in the defense of those scientists, they had no idea. All of it was magic. What we accept for as truth now is still magic in many ways, and so they were just trying to find the best, most rational explanation they could. Blending inheritance also proved to be incorrect, but these ideas such as germplasm theory, which is what it was referred to there, the idea that there were genetic sets of information in cells, we would call those chromosomes now, certainly panned out. Cell theory, that life is made up of discrete individual cells that are stuck together in tissues, and cells derive from other pre-existing cells. That's true. And, of course, the Mendelian rules of Mendelian genetics, which govern very simple inheritance patterns, are also known now to be true. 18 years after Mendel died, in the year 1902, a scientist named Walter Sutton made a remarkable connection. He had the insight to realize that the genetic information that was responsible for Mendelian inheritance was contained on chromosomes. Chromosomes had been seen before. A few years earlier, chromosomes were observed being divided during cell division. Scientists could see that the cell was going to an enormous amount of trouble to deal out these structures. Before one cell divided into two and a mother cell turned into two daughter cells, the cells were going through great pains to make sure that daughter cell 1 got one of these chromosomes and daughter cell 2 got the other. Daughter cell 1 got one of these chromosomes and daughter cell 2 got the other. And so Sutton realized, why in the world would cells be going to so much trouble to accurately and faithfully deal out these chromosomes to daughter cells during division unless they were important? Sutton called the individual units of heredity on these structures genes, and he proposed that there were many, many, many genes on each chromosome, and he was right. At the same time, another scientist named Thomas Hunt Morgan identified the first genetic mutant, and it was that discovery of mutations, he happened to be using flies, it was that discovery of mutations that made the connection between changes in genetic information giving rise to changes in traits. On top of, or standing above inheritance, mutations could also change individuals if it affected this genetic information. In the 1940s, so now we're about 30, 40 years after the time of Sutton and Morgan, we began using model organisms such as bacteria and viruses to learn even more, and this kind of led to the modern genetic revolution. Remember, simple model organisms for simple, basic questions. Bacteria and viruses for the fundamental principles of life. It's also around this time frame that DNA was characterized as the genetic molecule. I hope you know who's responsible for that discovery. That was Watson and Crick's hallmark contribution to the field. By 1966, about 10 years after that, the genetic code is determined. This is the translation dictionary, the molecular translation dictionary that allows us to understand how DNA sequences and genes give rise to proteins. It's not really well known, but Francis Crick of Watson and Crick is also responsible for the determination of the genetic code. Remember, DNA is an instruction manual, nothing more. So what this discovery in 1966 allowed was for all of us to understand how DNA was an instruction manual, how the information in DNA was encoded, and how that information was used to build proteins. It's the proteins that do everything for life, and DNA is nothing more than the instructions for building those proteins. In 1966, we learn how DNA contains the information for building proteins. Fast forward about 29 years. In 1995, the first genome is sequenced. It's that of H. pylori, a bacteria that causes stomach ulcers. 1 year later, remarkably, the first eukaryotic genome is sequenced. That's of Baker's yeast, Saccharomyces cerevisiae. 4 years after that, the rough draft of the human genome is complete. 3 years after that, in 2003, I'm already a graduate student at this time, the final draft of the human genome is released, and we are well on our way to breakthroughs such as personalized medicine, advanced genomics, genomic modifications, recombinant DNA technology, etc., etc. This doofy-looking geek over here is Watson, Jim Watson, when he was responsible for the elucidation of the structure of DNA. That's in 1953, 1 year after my father was born. In 2013, Jim Watson is still with us, although he is quite frail and old, and I'd like you to think about that for just one moment. In a single human lifetime, remember my father was born in 1952, and my father, of course, is still alive now. In a single human lifetime, we went from understanding what DNA was to all that we know now. All that we know now. In a single human lifetime. Even more astounding, Jim Watson himself, regardless of how big a jerk he might be, was responsible himself for the discovery of DNA as a molecule and still lives today to see all that came of it, including the entire sequencing of the human genome. So we've gone from understanding DNA as a molecule to understanding how DNA works as the instruction manual for building proteins, to understanding how mutations change the behavior of cells, leading to things such as cancer, to understanding how we might manipulate that DNA to give rise to cancer in laboratory organisms, or maybe even someday fight cancer in those same organisms. Everything we understand from life now stems from genetics and DNA at its backbone. All of the things that insult us, all of the things that make us sick, all of our responses to these environmental insults, these environmental situations, have underpinnings in genetics. And that genetics has come about in a single lifetime. Just amazing to me. All right, so let's wrap up. Some big concepts to know as we move forward through the material. Genetics is made up of 3 major subfields, and we will be touching on all 3 of them. It's important that you be familiar with them and know what they're called, and in general know what they contain. Transmission genetics is what we would refer to as classical or Mendelian genetics. The subfield of this transmission genetics includes the basic principles of inheritance that Mendel discovered and focuses largely on how traits are passed from parents to offspring on chromosomes. The second subfield is molecular genetics. That's my field of expertise in genetics, my passion. Here we're more interested in the biochemical nature of the gene molecule, of DNA. We ask questions like, what does the DNA molecule do? How does the cell manipulate it? How does the DNA molecule hold information? How do the DNA-dependent processes, such as transcription, translation, replication, gene regulation, DNA repair, how do they operate at the protein level? That's molecular genetics. Population genetics is much more relevant to the forensics field. Here we look at heritable patterns in entire populations. What percentage of the population has albinoism and why? What percentage of the population is prone to alcoholism and why? What genes can we predict are associated with these traits? How do these traits change with time? How do they change with location? How do they change with environmental stress, etc.? We look at the entire population of a species and ask those types of genetic questions of inheritance patterns. Essentially, you can say that population genetics is the study of evolution, how large populations within a species behave over time genetically. So you can represent these subfields in very simple ways. Molecular genetics looks at the molecule of DNA and the proteins that work with it, getting down into that molecular level as though you're in the nucleus watching these things happen. Population genetics deal with large populations of a single species and ask questions like, what type of genetic units, what type of genes can we expect to be responsible for the spotting patterns on the backs of ladybug shells? And transmission genetics looks at direct inheritance patterns between parents and offspring. So why does this mommy plant have one kitten that looks just like her, one kitten that looks quite a bit like her, and then 3 other cats that really don't have her black patterning fur at all? What is it about her genes and the genes she passed on to her children that allow them to either resemble her or not? That's transmission genetics, the transmitting of genetic information into the next generation. And plus, they're so cute. Other key things to keep in mind. The gene, as we've already said, is the fundamental unit of heredity. A single gene is the instructions for building a single protein, usually. There are multiple forms or flavors of genes that we refer to as alleles. Katy Perry is one of my kids' favorite singers. My iPod is chock full of Katy Perry songs, for better or for worse. I'm not going to lie, I enjoy them too. They're catchy, and I look forward to the Katy Perry hour right before dinner at home myself. But in any event, you could probably in some vintage record store somewhere find Katy Perry songs on vinyl, on an LP. I'm sure that they've created a select few for collectors. Cassette tapes aren't in wide use anymore, but there are still a few cassette players out there, and there's nothing stopping you from taking a Katy Perry song and putting it on a cassette tape. Of course, CDs are still, at least in some use, so we can easily find a Katy Perry album on a CD, I'm sure. And the medium of choice for all of us now are MP3s, and so we can find a Katy Perry song on MP3s. This is an analogy for alleles. The gene is Wide Awake. Okay? The Katy Perry song Wide Awake. That's the gene. That's the information. That's the content. The allele is the form that information takes. Wide Awake on vinyl, Wide Awake on cassette, Wide Awake on CD, Wide Awake as an MP3. Same information, same content, but the flavor, the form, the version is different. So the genetic example, hair color is the gene. That's the information. That's the content. That space on a chromosome is designated for hair color. So what then are the alleles? The alleles are black hair, brown hair, blonde hair, red hair. Okay? So it's important that you know the difference and similarities between genes and alleles. Genes confer phenotypes. A phenotype is how an individual looks. Phenotypes are traits. So genes give you a hair color. The genes that you have, the alleles that you have for hair color are your genotype. But they give you the hair color that you have on top of your head, which is your phenotype. The phenotype is the physical manifestation of your genes. You have blonde hair because your genome, your genotype, the DNA that you have, gives you blonde hair. But blonde hair is the trait. That is your phenotype. Chromosomes are long DNA molecules that are simply just staggered sets of genes. Genes are located on chromosomes. You have 48 chromosomes in each of your cells, responsible for about 20,000 genes. So we have DNA. That DNA is wrapped tightly and compacted into a chromosome, but all along that DNA are genes. There's a gene here, there's a gene here, there's a gene here, etc. You have 46 chromosomes in you. 23 of those came from your mother, 23 of those came from your father. Each of us are truly half of each our parent. Our parents contribute half of our genomes to us, and that makes us the unique individual that we are. Chromosomes replicate, divide, and separate through two different cellular processes, one called mitosis, where a single mother cell divides into two genetically identical daughter cells. The other is called meiosis, where a single mother cell gives rise to 4 daughter cells that have half of the mother cell's DNA content. This is used for straightforward cell division. Meiosis is used for sexual reproduction. Daddy contributes one of his half cells. Mommy contributes one of her half cells. The two halves come together to make a whole. I've already said it a number of times, but genes are the instructions for building proteins, no more and no less. The central dogma of molecular biology says that a single DNA gene is transcribed into a single RNA molecule, which is then translated into a single protein, and the protein goes and does the job the cell needs done to live. It's all about the proteins. It's all about the proteins. So why do we need DNA at all? Well, DNA is the instruction manuals for those proteins. Get rid of the DNA, the cell has no idea how to make the proteins it needs to survive. So what did we talk about in this lecture? Well, we started off by saying there's no way around it. Tough noogies. You got to take genetics because genetics is rapidly becoming the backbone of all biomedical fields, including neuroscience, forensics, personalized medicine, etc. The human genome consists of about 20,000 genes spread across 46 chromosomes. Each of these genes is the instructions for building a single protein that is the central dogma of molecular biology. Every living thing on this earth uses DNA in the same way as the instructions for building proteins. That means by studying how one living thing uses DNA, we are studying how all living things use DNA, and that allows us to use model organisms to learn about ourselves. We talked about bacteria, yeast, worms, flies, mice, and even plants, all the lowest form possible answering the questions we want to answer, all giving us information about how we work as human beings. As people, we've understood and manipulated genetics for thousands of years. We've been selectively breeding our own foods for thousands of years without even understanding what we were doing. We then went on to the actual field of genetics. Gregor Mendel is the father of Mendelian genetics. He opened this field up. Transmission genetics stems directly from his findings. Sutton proposed that genetic information was contained in and or on chromosomes, and he was right. And those two connections, understanding the inheritance pattern in general, and where in the cell those inheritable molecules existed, set us off on our way. We covered some of the other milestones in the history of genetics. I won't rehash them here. We talked about the subfields of genetics, transmission or Mendelian genetics, molecular genetics, and population genetics. In that conversation, we kind of skirted around the difference between prokaryotic and eukaryotic cells, but I'm sure you know that already. We touched on the definition of alleles. Alleles are different versions of genes. Blue eyes, brown eyes, hazel eyes are alleles for the gene of eye color. And I'll stress it one more time, the central dogma of molecular biology sits below all that we will discuss this semester as our backbone. Why DNA exists and why DNA is important is the central dogma of molecular biology. The next lecture that we watch is going to be a long one. It's actually going to be 2 lectures in one. So next week you will only have this one big lecture to watch as opposed to 2 separate lectures. The name of the chapter and the name of the lecture is technically Chromosomes and Cellular Reproduction, but that's really just code for the processes of mitosis and meiosis. We need to discuss how cells divide and divvy up their genetic information, their chromosomes, before we can actually talk about what those chromosomes do. So we'll get that out of the way first, and then launch our way into true genetics. Until then, and as always, thanks for watching, And I'm looking forward to seeing what you guys thought of this on your optional extra credit reflections so that we can fine-tune this a little bit and maybe make this an enjoyable experience for everyone. See you guys soon.